Welcome to another episode from the Veterinary Research Channel. Today, we're diving deep into a fascinating study that explores a common yet mysterious condition in our canine companions, Fibrocartilaginous Embolic Myelopathy, or FCE. This condition, as we'll discover, has some intriguing links to genetics and breed predispositions. FCE is a disease that has puzzled veterinarians for years. It occurs when fibrocartilaginous material, essentially tissue from the intervertebral disc, enters the vascular system and blocks blood flow to the spinal cord. This blockage leads to a range of neurological symptoms, often appearing suddenly. Think of it like a stroke in the spine. While it's frequently seen in larger breeds, smaller and medium-sized dogs aren't immune. Our story begins with a team of researchers from several veterinary institutions across the United States and even Finland. They set out to investigate a possible genetic connection to FCE, focusing on something called the FGF4L2 Retrogene. This gene is known to be associated with chondrodystrophy, a condition affecting cartilage development, leading to shorter limbs and sometimes premature degeneration of the intervertebral discs. The central question was this: is there a link between this chondrodystrophy-associated gene and the occurrence of FCE? The researchers hypothesized that dogs with the FGF4L2 Retrogene might be less likely to develop FCE. This idea stemmed from the observation that FCE is rarely reported in chondrodystrophic breeds. To get to the bottom of this, the team embarked on a retrospective, multi-center study. They gathered data from 98 dogs diagnosed with FCE through histopathology, which means the diagnoses were confirmed by examining tissue samples under a microscope. This is a critical point because many conditions can mimic FCE, making accurate diagnosis essential. The researchers extracted DNA from tissue samples to genotype the dog for both the FGF4L2 and FGF4L1 retrogenes. FGF4L1 is another retrogene associated with chondrodysplasia, a different form of skeletal dwarfism. By looking at the presence or absence of these genes, they hope to find some statistical correlations. They also gathered historical breed data from previously published FCE cases to compare with their findings. Statistical analysis was then performed to determine if there were any significant associations between breed, FCE, and the presence of these retrogenes. So what did they find? Well, the FGF4L2 genotype was successfully determined in 89 of the FCE cases. Interestingly, FCE was present in 22 dogs from breeds that commonly carry the FGF4L2 gene, but all of these dogs were wild type, meaning they did not have the mutated gene. Only 2 Labrador Retrievers with FCE actually carried the FGF4L2 allele. When they compared the frequency of the FGF4L2 allele in FCE cases to the predicted frequency in the general hospital dog population, they found a significant negative association. In other words, the FGF4L2 allele was underrepresented in dogs with FCE. The study also revealed that certain breeds were overrepresented among the FCE cases. These included boxers, Great Danes, Yorkshire Terriers, Bernese Mountain Dogs, miniature Schnauzers, Rottweilers, and Shetland Sheepdogs. This suggests that these breeds might have a higher genetic predisposition to developing FCE. Now, let's put these findings into perspective. The fact that FCE is uncommon in chondrodystrophic breeds, which often carry the FGF4L2 retrogene, supports the idea that there's something about their biology that protects them from this condition. The researchers suggest that FGF4, the gene influenced by these retrogenes, plays a role in angiogenesis and vascular integrity. So what does this mean? The study data suggests that the historical observation that FCE is uncommon in chondrodystrophic dog breeds is supported. It appears that the genetics that cause shorter legs also somehow protect against spinal strokes. This protection may have to do with the nature of the intervertebral disc or the structure of the blood vessels around the spine. Specifically, anatomical differences between chondrodystrophic and non-chondrodystrophic dogs might provide clues into how FCE develops. Let's consider the implications. This study reinforces the importance of accurate diagnosis in FCE cases. Many conditions can mimic FCE, and confirming the diagnosis through histopathology is crucial for research and treatment purposes. The breed predispositions identified in this study can help veterinarians assess risk in certain patients. Knowing that boxers, Great Danes, and Yorkshire Terriers might be more prone to FCE can lead to earlier diagnosis and intervention. One of the most intriguing aspects of this research is the potential for future studies. The researchers suggest that comparing the intervertebral discs and spinal cord vascular anatomy of chondrodystrophic and non-chondrodystrophic dogs could reveal why the former are less susceptible to FCE. Perhaps the premature degeneration of the nuclear material in chondrodystrophic breeds makes it less likely to enter the vascular system. Or maybe differences in vascular development, influenced by FGF4, play a protective role. The possibilities are numerous and warrant further investigation. Of course, this study isn't without its limitations. The number of confirmed FCE cases was relatively small, which can affect the statistical power of the findings. Also, breed predisposition was assessed using population data from a single institution, which might not fully represent the broader dog population. Despite these limitations, the study provides valuable insights into the genetics and breed predispositions associated with FCE. It also opens up new avenues for research into the pathogenesis of this perplexing condition. In conclusion, this study sheds light on the complex interplay between genetics, anatomy, and disease in our canine friends. By identifying a negative association between the FGF4L2 Retrogene and FCE, the researchers have provided a foundation for future investigations into the underlying mechanisms of this condition. Understanding these mechanisms could lead to better diagnostic tools, preventative strategies, and ultimately, improved outcomes for dogs affected by FCE. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.